In December 2019, the world was awakened by a pneumonia disease outbreak with the first case detected in Wuhan, China. The Hunan market in Wuhan was suspected as the source of the pandemic. This disease was identified as a novel strain of coronaviruses SARS-CoV-2, and later, the disease was decided to be termed COVID-19. COVID-19 most often causes respiratory symptoms that can feel much like a cold, a flu, or pneumonia. COVID the 19th of May attack more than the lungs and respiratory system. Other parts of the body may also be affected by the disease. This virus is transmitted when people with COVID-19 breathe out or cough, expelling tiny droplets that contain the virus. These droplets can enter the mouth or nose of someone without the virus, causing an infection to occur. The disease was rapidly spread globally, causing a high number of infections and deaths all around the world, which led the World Health Organization, WHO, to declare the disease a pandemic. Rapid transmission of disease does not only threaten public health but also the global economy. Due to that, vaccines and drugs are most needed to combat this outbreak as soon as possible. Although, the development of highly effective and safe vaccines and drugs is very challenging and mostly takes years before making it available to the public. With the advancement of the technology, computer science, and data available on disease, especially on genome data of SARS-CoV-2, the application of bioinformatics tools in the development of the COVID-19 vaccines and drugs candidates become useful, and at the same time, gives a lot of advantages to the researchers in terms of cost and time reduction. But first, what is bioinformatics? Bioinformatics is an interdisciplinary scientific field involving the application of technology and computer science to gather, store, organize, and analyze biological data, which are DNA, RNA, and amino acid sequences. Bioinformatics helps scientists acquire information from biological data for a better understanding of the physiological and pathological mechanisms that happen in an organism. Now, let's see how bioinformatics is being applied in vaccine and drug design for COVID-19. In vaccine design, 3 approaches were done, which are reverse vaccinology, Immunoinformatics, and structural vaccinology. Reverse vaccinology is an approach that helps in the recognition of possible target antigens by analyzing the genome of the pathogen. In detail, this approach will use the software or tools to analyze the genome and identify the genes that encode the antigen. Then, the genes will be further analyzed to determine the physical and chemical characteristics of the antigen. This helps in the study and identification of the antigen that can be used in vaccine development. Moreover, this approach too helps in the study of the pathogen. Next, the Immunoinformatics approach helps study the immune system of an organism, analyze genomic data to study the immune response towards specific antigens, and determine potential antigens from the viral genome that are able to mount an immune response. Among the tools that are being used in this approach are TIA-Predict, CTL-Pred, NetMHC, and EpitopeMap. Adding on, machine learning algorithms were also applied in this approach to aid the prediction of immunogenic molecules. Immunoinformatics approach has a lot of benefits, as it can fasten the identification of immunogenic molecules and at the same time improve safe vaccine development. The last approach in vaccine design is structural vaccinology. Structural vaccinology functions to predict and analyze the structure and the conformation of the antigen for the candidate vaccine, as it can affect the antigenicity of the antigen. This approach is important in determining the stability and effectiveness of the antigen structure that can be used in vaccines. Several techniques are used in this approach, which includes molecular docking, dynamic simulation, and homology modeling. In spite of the various functions of the application of bioinformatics in predicting vaccine candidates, it also presents several limitations. First, bioinformatics tools are unable to determine the suitable animal model that can be used in the testing of vaccine candidates. Secondly, bioinformatics tools can only identify and analyze the protein as a molecular target compared to the conventional approach that is able to identify other molecular targets such as polysaccharides. The next limitation is that bioinformatics tools allow the identification of the hidden or not accessible in vivo antibodies epitope that leads to the rejection of the candidate vaccines, which leads to a waste of time and money spent. Other than that, due to the pleomorphism of MHC class I molecules, a lot of data and information is needed to train the prediction tool for designing the vaccine. However, with the rapid need for the COVID-19 vaccines, less time was spent on the production of datasets, which can reduce the precision of the prediction tools in designing the vaccines. Lastly, incomplete genomic data for underdeveloped and developing countries may cause a reduction in vaccine efficacy for this population. Examples of machine learning that are being used in antigenic peptide prediction for the development of vaccines are SPRINT, Model AMP, PEPATTRACT, PIGS Pro, etc. For the drug design, drug repurposing was chosen over the discovery of new drugs as it can reduce the time and cost, and most importantly due to its certified safety. To apply in silico method in drug repurposing, the drug target and its protein structure or its close homologue must be first identified. After that, bioinformatic tools will be used to screen and identify the suitable existing drugs that are able to interact with the target molecule. In the case of COVID-19, Pro of SARS-CoV-2, which is the protease enzyme that plays a role in viral replication and transcription, has been detected to be a good drug target. Then, the three-dimensional, 3D structure of the enzyme was determined. Bioinformatics tools such as molecular docking and dynamic simulations were then applied to identify the drug candidate. Drug candidates that have been identified by using this approach are lopinavir and remdesivir. Other than that, the network bioinformatics approach has also been applied by scientists. This approach involves phylogenomics analysis of the SARS-CoV-2 genome with other closely related viral families that have similar structure and function of the protein for identification of the drug target. After that, a drug prioritization algorithm was used. Coronavirus Explorer, COVAX, is one of the tools used in this approach. COVAX integrates the information of human protein-protein interactions, human virus-protein interactions for SARS-CoV, SARS-CoV-2 and drug-protein interactions into a large-scale interactome, which can aid in the recovery of drug targets, drug candidates, and study the interaction of drug candidates and drug target. From this approach, 30 candidate drugs that can be used against COVID-19 were identified. Some of them are thalidomide, pseudoephedrine, andrographolide, and abacavir. Among machine learning tools used for drug candidate designing include CHAIMM, AMBER, DOC, Zinc Pharma, Gandhi, Catalyst, HYDE, and LUDI. Overall, advancement in technology allows bioinformatics tools to replace conventional methods in the research of COVID-19, which enable faster response and management of the disease, especially in the development of drugs and vaccines. Besides, compared to conventional methods, the usage of bioinformatics tools in the study of the COVID-19 and the development of drugs and vaccines is more economical and affordable for low-income and middle-income countries. This can speed up the vaccination rate throughout the world, hence, being able to eradicate the disease sooner than expected.